Today's topic for discussion is a practical protocol of mycology. And today's practical is the transverse section of gill. Gill is a membranous structure of a fruiting body of Agaricus. It occurs in a fruiting body of Agaricus, which is known as Basidial. And let us see the internal structure of gills by taking transverse section. And what we require for this practical, the requirement is the gills of Basidial of Agaricus, needle, brush, blade, slide cover slip, cotton glue, microscope and blotting paper. These are the requirement of practical purpose. Okay. And what we have to do, take many sections through the membranous gill. Gill will be provided to you in the examination or during practical, and that membrane like structure, there are many membranous structures occur in a fruiting body of Agaricus, and that membranous structure, it is known as gill, and that gill you have to take a thin section of that gill and mount a thin section in a drop of mixture of cotton glue and lactophenol and observe this slide under microscope. Let us see the internal details what we can see under microscope. The transverse section of gill shows the following details, that is the central layer, that is trama, which is known as trama. It is the central region of the gill that consists of loosely arranged hyphae which are called trama. And this trama accumulate more moisture because the hyphae are arranged loosely and there are many interhyphal spaces left. And all the hyphal threads present in this region, they are sterile, hence trama is supportive in function. It is a supportive region of the gill. Okay. And on both the side of the trama region, subhymenium is present, and there are few layers of the subhymenium is made up of few layers of compactly arranged hyphae and situated on both the side of trama, and such a compactly arranged hyphae on both the side of trama is described as subhymenium. Again, the subhymenium is also supportive in nature. It supports the gill and it is also a sterile region. But in comparison with the trama, the interwoven hyphae of subhymenium, they are somewhat compactly arranged. Next. And the subhymenium produces layers of compactly arranged fertile hyphae on both the sides, which is known as hymenium. In fruiting body of Agaricus, the hymenium, it is a fertile layer and it bears a series of club-shaped structures which is called Basidial Layer. And Basidial Layer, again it develops on both the sides of the subhymenium, and Basidial Layer is developed from the hymenium. The hyphae of hymenium, they are fertile hyphae, and in presence of enough food material, the Basidial Layer is developed. And each basidium is a club-shaped structure which produces 4 pointed structures at its tip, and these pointed structures are known as sterigmata. The sterigmata are pointed and these are sterile structures that gives mechanical support to the basidiospores. Basidiospores are produced at the tip of each sterigmata. And how is the basidial layer? The basidial layer consists of a series of basidia. Basidia are club-shaped or finger-shaped structures. Just I described. At the tip of each basidium, 4 sterigmata are there, and on each sterigmata, single basidiospore is developed at the top of sterigmata. Sterigmata, it is a sterile region, sterile pointed knob which is developed by the tip of the basidium. Each basidium is having 4 sterigmata and 4 basidiospores are developed at the tip of sterigmata. And the basidiospores which are developed at the end region of sterigmata, the basidiospores are very small in size. They are spherical, unicellular, uninucleated, soft-walled structures. And morphologically, all these 4 basidiospores of each basidium, They are similar in size and shape, and morphologically they are similar, but at physiological level and at genetical level they are different. Out of 4 basidiospores, 2 basidiospores are of positive strain and 2 are of negative strain. The basidiospores germinate on soil surface containing high quantity of humus. Humus in the soil favors the growth of the Agaricus, and many the fruiting bodies are developed out of the soil surface. On germination, what happens? The basidiospores produce two types of hyphae, because the positive strained basidiospores produces positive strained hypha, and negative strained basidiospores produces negative strained hyphae. And these two types of hyphae, positive strained and negative strained, they are present just below the soil surface, and by chance the somatogamy occurs below the soil surface. After somatogamy, again dikaryotic hypha is developed, from which many the fruiting bodies are developed in the favorable environmental condition. Okay, this is all about the internal structure of transverse section of gill, and this is the photograph shown in the slide. That is, central region of the gill is trama, and both the side of trama that subhymenium is there, and on both the side of subhymenium that produces hymeneal layer. Hymeneum produces again basidial layer, and basidia produces sterigmata. On the sterigmata, each basidium produces 4 basidiospores, and 2 are of negative strain, 2 are of positive strain. This is the details of transverse section of gill. Next. Thank you very much. Save trees, save earth.